So by way of review, last time we were talking about some of the differences between prokaryotic and eukaryotic genes. And we said that prokaryotic genes do not have introns like eukaryotic genes do. And remember, in eukaryotic genes, the initial messenger RNA transcript has to have the introns removed, right, to make the final functional messenger RNA. And we said that this presents a problem for cloning a eukaryotic gene into a prokaryotic cell, because if you take this gene and put it into a bacterial cell, it's not going to form a functional product, because this is the messenger RNA that's going to be translated to make the protein. And because of the introns, there's going to be a lot of extra amino acids. And so that protein is not going to function. So we said what we had to do was isolate the processed messenger RNA transcript with all the introns removed, use the enzyme reverse transcriptase to make a DNA copy. Now we have a clean gene, no introns. And then we can insert that gene into the bacteria. And we're not going to get to it today. We'll have to finish it up on Wednesday, but we'll talk about the process of transformation, whereby we can insert pieces of DNA into bacterial cells. We also said that prokaryotes can produce a protein faster, because in eukaryotes, the messenger RNA is made in the nucleus and then has to be transported to the cytoplasm, where it can hook up with a ribosome and be translated to make a protein. But in prokaryotes, there's no nucleus, no nuclear membrane. So transcription and translation occur simultaneously in the cytoplasm. So as the free end of the messenger RNA transcript, it can hook up with a ribosome even before the entire messenger RNA has been transcribed. And so translation can occur along with transcription. And that allows bacteria to make proteins faster. So when it comes to translation, we're not going to go into all the same detail you learned in Biology 150. We're just going to review some basics. But what students always remember from 150 is that the packets of information are encoded by the triplet codons, or just codons for short. So groups of 3 nucleotides. So students always remember codons. And remember, there are some special codons like AUG, which is the start codon. So all proteins start with AUG. And there are 3 stop codons, UAA, UAG, and UGA. So the ribosome needs to know where to start translating, and that's AUG. And the ribosome needs to know where to stop translating, and that's one of those 3 stop codons. So every biology student should be familiar with the genetic code. And does this look familiar to everyone? Yes?
Yes.
So Bryant, do you know how to use it? So remember, in RNA, there is no T, it's U, right? And so what you need to be able to do, if I give you a piece of DNA, you need to write me the complementary messenger RNA sequence. And then you must also be able to use the messenger RNA sequence to find the amino acid sequence. Now, I don't expect you to know what all the amino acids are. I mean, that would be nice, but I don't expect you to know them. But let's say that you have this codon. Let's see. Oops, switch to right here. So what amino acid goes with that codon? And so remember, you take the first nucleotide, the first base, you look on the left-hand side, and then you take the second nucleotide, or the second base, and go across the top, and then triangulate. So we find the C here, and go across the top to A, and come down, right? C and A and CAG, and that codes for glutamine. Right, so is all that familiar? Yes. What if I gave you AGA? What amino acid does that code for? Arginine, right? So did we all follow? Always use the first base on the left, second base go across the top, and then triangulate to find the third base. So you should be able to give me the amino acid sequence to an RNA sequence. And we're not going to worry about anticodons or any of that fancy stuff. I'll just give you the sequence, assume it's in the proper orientation, write the complementary strand, and go from there. So any questions? I have questions. Yeah, Christiana?
Please, Professor, can you explain this, the CAG again, please?
So again, this is the codon CAG. Look, the first base C you find along the left, and there's C there. And the second base A, you go across the top to A, and then you triangulate. And then you find the third base, CAG, which codes for glutamine. For AGA, same thing. You find the A on the left, G across the top, and come down and triangulate. And then you find AGA here, and that's arginine. So is that good, Christiana?
Thank you.
Now, I have some questions. So how many naturally occurring amino acids are there? Has anybody taken nutrition yet? How many amino acids are there? $20, right? 20. So we need 20 codons. I see a lot more than 20 codons here, right? I mean, for example, leucine, LEU. How many codons code for leucine, LEU? You sure, Bryant? Leucine? See, there's 6 for leucine. How about serine, SER? Six as well, right? You got 4 here and 2 there. So the idea is that for almost every amino acid, there's more than one codon. The exception is methionine. AUG is start and methionine. But for almost every other amino acid, there's more than one codon. So the genetic code is redundant. And the question is, why is it redundant? If we only have 20 amino acids, why do we need 64 codons here? I mean, there's 64 codons. Obviously, we need at least one start and one stop, but that would only be 22 codons. So why is it we have 64 when we only need at most 22? Today is a math day. We got lots of math, especially in lab. So we know we have 4 nucleotides, right? ATGC. And if each nucleotide encoded for 1 amino acid, that would just be 4 amino acids, right? 1 for each nucleotide, so that's no good. But what if we took those 4 nucleotides and we put them in pairs? How many combinations do we get when we do that? Right, so we have 4 nucleotides and we're going to pair them. All right, so 4 squared. How many combinations is that? $16, right? But that's not enough, right? We have at least 20 amino acids, so putting our nucleotides in pairs is no good. So what if we put them into groups of 3? Then how many combinations do we get? What's 4 times 4 times 4? 64, right? Well, that's how many codons we have, 64. It's a lot more than we need, but it's the only way to arrange 4 nucleotides and get at least 20 unique combinations. So that's why they're grouped in 3. So that's why we have the triplet codons. That also has the benefit of providing a little bit of flexibility, a little bit of protection against changes in DNA. And what do we call those changes in DNA? What do we call changes in the genetic material? Mutations, right, Taylor? So if we go back to the leucine here, as long as you have a C and a U, it doesn't matter what that 3rd nucleotide is. You still get leucine. So you can have some mutations occur which don't actually change the codon in a way that changes the amino acids. So there is a little bit of, cushion, protection, flexibility against some mutations. But any questions on how we did this? So I'm not going to actually go into any of this stuff here. We're going to move on to the regulation of bacterial gene expression. So as you know, bacteria are small single-cell microorganisms. They have a certain number of genes, and all of those genes are not going to be turned on. So bacteria need to be able to turn certain genes on when they're needed and turn them off when they're not needed. And so bacteria need a way to regulate their gene expression, because if every gene is turned on and being transcribed and translated and making proteins and enzymes, if they're not needed, that wastes energy. So bacteria cannot afford to be wasteful. But there are certain genes that code for certain enzymes that are so important, they're always needed. We call those genes and enzymes constitutive because they're always needed. So those genes are always turned on, always being transcribed and translated. For example, the genes for glucose metabolism are constitutive because most bacteria are either going to perform aerobic cell respiration or fermentation. So they need to be able to use glucose. So those genes are always turned on. But for most everything else, the genes need to be turned on and off as needed. So they need to be induced or repressed. Induction is the process of turning on a gene that's normally turned off. Inducible genes are typically those that code for enzymes needed for catabolic pathways, for those pathways that break down a food molecule and use it to make ATP. So if a different food source is required, then many bacteria can turn on a gene needed to make enzymes so they can use that alternate food source. But if that alternate food source is not required, those genes are turned off. Otherwise, it would be wasteful. When it comes to repressible genes, those are genes that typically code for enzymes for biosynthetic pathways, so for making things. And here again, the idea is once the cell has made enough of a particular product, it wants to stop making that product. And so the gene gets repressed. So normally a repressible gene is on and needs to get turned off. Inducible genes are off and need to get turned on. Now, we already learned about one way that bacteria can regulate the formation of product. What was that way? How can bacteria regulate the formation of product?
Is it by operon?
No, something we already talked about. We talked about a way that bacteria can regulate the formation of product. What was that mechanism?
Is it biosynthesis?
No. When the bacteria has made enough of a product, it wants to stop making more. We talked about a mechanism. Nobody's remember? Taylor, you don't remember? Christina? Bryant? Nope. That's what we talked about before today. How soon they forget. Do you remember where the end product can feed back to an enzyme, bind to the allosteric site, and change the shape of the enzyme and turn it off? What did we call that? Well, be more specific, Bryant. Feedback inhibition, correct, Sarah. So cells already had one way, right? And the idea was to turn an enzyme off and shut down the pathway in order to save energy, right? Here, the end product can actually feed back to the gene, turn the gene off so it's not transcribed or translated. And that saves a lot more energy, right? Because the cell's not making RNA, it's not translating it to make protein. So cells have two ways to regulate, at least in prokaryotic cells. But when it comes to gene regulation, bacteria and other prokaryotes use the operon. And so operons are almost exclusively found in prokaryotes. There are some examples in yeasts, which are eukaryotic single-cell microorganisms, but us and other complex organisms, we don't use operons. We have many different mechanisms for regulating gene expression. And it's very, very complicated for eukaryotes. But for prokaryotes, it's the same general idea, right? The operon model. Although not all operons work the same way, some of them are actually very complicated, but we're going to look at the general idea behind operons. So, of course, all genes have a promoter. Remember, the RNA polymerase binds to the promoter, And of course, they have all the genes that are actually going to be transcribed and then translated. But for the operon, there's this intervening sequence called the operator sequence. This is where the regulation occurs. And as we'll see, a repressor protein binds to the operator sequence, and that acts like a physical barrier and blocks RNA polymerase from getting past. And in that way, these genes are turned off, right? Because RNA polymerase cannot get past that repressor protein and transcribe those genes. Now, there is a gene that codes for the repressor protein. It's located some distance away from the operon, and it's under its own control mechanism, because you can't have a regulatory gene in the operon because that just wouldn't work. But that's the main idea. Now, here we also see another important difference between prokaryotes and eukaryotes. In eukaryotes, they have one promoter for one gene. But you notice here, we actually have multiple genes under the control of a single promoter. So again, that's very efficient. The idea here is that all those genes code for enzymes needed for the same metabolic pathway. So all those genes get turned on and transcribed and translated at the same time. Eukaryotes, you just tend to have one promoter for one gene, but prokaryotes have one promoter for multiple genes that are all needed for that metabolic pathway. So again, that's very efficient to put all those genes under the control of the same promoter, so they all get transcribed and translated at the same time, so all the enzymes are available for that metabolic pathway. And there is a name for that, and that name is polycistronic. So polycistronic. So you have an RNA that actually has multiple genes on it. So when it's translated, multiple enzymes will be made. But eukaryotes normally just have one promoter, one gene. So that's monocistronic. So that's another important difference between prokaryotic and eukaryotic DNA. So here we see a repressor protein is bound to the operator. So when RNA polymerase hops onto the promoter, it's blocked. It cannot get past that repressor protein. So this operon is turned off. So we're going to see an example of an inducible operon and see how that gets turned on. And the classic example is the lactose operon, or the lac operon. So let's go back and revisit 150. What kind of saccharide is lactose? What kind of saccharide is lactose? It's easier to say it than to spell it. It's a disaccharide, right, Sarah? So a disaccharide. What are the two component monosaccharides that make up lactose? So what's lactose made out of?
Glucose and galactose.
Right, so glucose and galactose, right? So glucose and galactose. So where do we find lactose? Where do you find lactose?
Sugar.
No, it's a type of sugar, but where do you find it? in milk, right? In dairy products. So is lactose commonly found in the environment? Is lactose a common sugar in the environment? Yes or no?
Yes.
Really? Look around the room. You see milk everywhere? Look outside your window. You see milk out there? No. So it's not very common. So the lactose operon is normally turned off. It's only going to be turned on when absolutely necessary. Now, in that case, lactose serves as an inducer. And as we're going to learn, all the glucose has to be gone first. So once the cells use up all the glucose, an alarm signal actually goes off inside the cell, cyclic AMP, and the cell has to look around for something else to eat. If lactose is present, then lactose can induce the operon. And the way that works, there's a slight conformational change in the lactose. We're not going to worry about that. But lactose can bind to the repressor protein. When lactose binds to the repressor, it changes the shape of the repressor protein so it can no longer bind to the operator. And remember, as is almost always the case with everything, binding is reversible and concentration dependent. So the repressor protein is on the operator. Every now and then it comes off and then goes back on. But there's usually enough repressor protein to stay on the operator almost all the time. But if lactose is present and all the glucose is gone, lactose will bind to the repressor protein so it no longer binds to the operator. When RNA polymerase hops on the promoter, it's able to move past the operator sequence and start transcribing those genes. And there are 3 genes in the lac operon. The gene for beta-galactosidase, which is the enzyme that splits lactose into glucose and galactose. Permease, which dramatically increases the uptake of lactose from the environment. And transacetylase, which is not actually required for the cell to use lactose. And they've been studying this for decades. If you knock out the gene for transacetylase, bacteria have no problem using lactose. And so they think the purpose of transacetylase is that sometimes sugars, when they experience a conformational change, they actually become kind of toxic to the cell. And so transacetylase is believed to help deal with toxic forms of sugar. But it's not actually necessary for the cell to use lactose. But these 2 genes are. So E. coli happens to love lactose. Now, E. coli will use glucose first if it's present, and then it will look for something else to eat. If lactose is present, then the lac operon gets induced. It gets turned on, and the cell can start using lactose. But even if there's 1,000 times more lactose than glucose, E. coli will not touch the lactose. E. coli will use all the glucose first. Then it will look for something else to eat and then switch over to using lactose. So if you grow E. coli in a culture of glucose, you have a nice steep exponential growth here. If you just grow E. coli only on lactose, you still get exponential growth, but it's slower. It's not as steep. So the bacteria are not growing quite as well. If you grow E. coli in a mixture of glucose and lactose, the E. coli will use all the glucose first. Nice steep growth curve. And then they stop dividing and they enter into lag phase. What's happening during lag phase? All right, so they're growing great until they use up all the glucose. And then they enter into lag phase and stop dividing for a short period of time. So what are they doing? So what happens during lag phase? During any lag phase, what's happening? But what exactly are they doing during lag phase? Right? You were just tested on that. So what are they doing? They're sensing the environment, right? They know they're in trouble. They just ran out of glucose, so they got to look for something else to eat. So they're sensing the environment, and they see lactose is there. So now they start turning on the lactoperon and start synthesizing those enzymes so they can use lactose for energy. Once they're ready, they start dividing again, but not quite as well as they were on glucose. So once again, why is it that the bacteria will not touch the lactose until all the glucose is gone? Somebody give me something. So what do we know about the genes for glucose metabolism? I had this earlier. What do you know about the genes for glucose metabolism? So tell me something. It's going to be a long lecture if you don't answer. Roxy, have any ideas? Craig? So what can you tell me about the genes for glucose metabolism? What does that say? I'm going to wait till you guys talk to me. Right, so the genes for glucose metabolism are constitutive. So they're always being transcribed and translated. So those enzymes are always there. So if the cell were to turn on another operon and start making enzymes that it doesn't need, what would that waste? Right, so what does that waste? If you got to turn on genes that you don't need. Energy, right? Energy. So again, bacterial cells cannot afford to be wasteful. So as long as glucose is present, it's going to use all the glucose up first. Then it will look for something else to eat. If there's something else available, then it will turn on the operon in order to use that alternate food source. But that's why the bacteria don't grow as well on lactose, because they've had to turn on the lac operon. They have to transcribe and translate those genes and make those enzymes needed to use lactose. All that takes ATP, all that takes energy. And so the return on the investment is lower with lactose than it is for glucose. So does that make sense? I'm going to show you an animation of the lac operon. And the link is on my web page. So we're not seeing all the genes. We're just seeing the LAC-Z gene, which is the one that codes for beta-galactosidase. And so this is the normal state for the operon. It's normally off, right? The repressor protein is bound to the operator. RNA polymerase hops on, and it can't get past. So it can't translate any of the genes needed for to use lactose. So, but you notice every now and then the repressor protein comes off, and it can come off, and sometimes that allows the RNA polymerase to sneak in and transcribe a little bit. But most of the time, there is enough repressor protein that it will stay bound to the operator sequence. And so the operon is off almost all the time, right? But every once in a while, a polymerase can sneak in and get through. But normally, there is no enzymes for lactose utilization being produced. So how do we turn on the operon? Well, all the glucose has to disappear, and then the cell, it sets off an alarm signal, cyclic AMP, which helps to induce the lac operon. So we need to give the cell some lactose. And we have a lactose gun here, and we can fire off a bunch of lactose. And so the beta-galactosidase enzyme will split lactose into glucose and galactose. But remember, lactose can bind to the repressor protein. So the repressor changes shape and no longer binds to the operator sequence. And that will then allow the RNA polymerase to move past and transcribe all the genes in the lac operon. We're just looking at the beta-galactosidase gene. So that gene is transcribed, and then it will be translated to make the enzyme beta-galactosidase. So here transcription has occurred. Now translation to beta-galactosidase, which will then go and split all the lactose into glucose and galactose so the cell can use it. But eventually the cell does use up all of the lactose. And so once all the lactose is gone, it will no longer be available to bind to the repressor protein. And so once all the lactose is gone, it comes off the repressor, and the repressor will go back to its normal shape and bind to the operator sequence. And so now the cell has to look for something else to eat. So again, it will enter into a lag phase while it senses the environment to find what else is out there that it can eat. And it will turn on other operons as needed, right, to use those alternate food sources. So does that make sense? So that's the basic idea behind operons. And the lac operon is an inducible operon. If you took 150, you may recall that we also talked about the tryptophan operon, so a repressible operon. So the cell is making the amino acid tryptophan, so it continues to make tryptophan until it's got too much, and then the tryptophan can act as a co-repressor and turn off the tryptophan operon. So that operon gets repressed. So the tryptophan operon is a repressible operon. It's normally turned on and needs to get turned off. So the cell doesn't make any more product. And actually the cell stops making the enzymes needed for that synthesis. And that saves a lot more energy than feedback inhibition does. But there are many, many different operons. And they're all not exactly the same. Some of them are very complex. In an upcoming lab, we're going to talk about transformation, that experiment, and we'll just mention the arabinose operon, a very complicated operon. But it's all about energy, and a bacterial cell will not turn on genes that it does not need until it needs to. Otherwise, it's going to waste energy doing so. So does that make sense? So then on to mutations. As we already said, mutations are changes in the genetic material, and they can be neutral, beneficial, or harmful, although most mutations do tend to be harmful. It's actually possible for a mutation to be beneficial. That's not normally how we think about mutations, but in fact, every once in a while, a mutation occurs that confers or gives an organism a benefit, a selective advantage, and that allows that organism to survive better, reproduce more, and the offspring survive better and reproduce more, and eventually that mutation becomes part of that species. And so what do we call the accumulation of beneficial mutations leading to adaptation and survival of a species? What do we call that whole process, the overarching theme of biology? What is the accumulation of beneficial mutations leading to adaptation and survival? It's all important in biology. Evolution, that's it, right? So without all the bad mutations, we never get any good ones. And so we have to take the bad with the good in order to get any beneficial mutations at all. And mutations can and do occur spontaneously. DNA polymerase is not perfect, right? Mistakes happen. But if you want to increase the rate of a mutation, you need a chemical or physical agent to do that. And we call that chemical or physical agent that increases the rate of mutation a mutagen. We'll talk about a couple of those in a few minutes. But some types of mutations. When it comes to base substitutions or point mutations, we're just changing one nucleotide. And that one nucleotide changes the codon. And so if that codon changes to a different codon, which changes the amino acid, we call that a missense mutation. In this example, the codon GGC codes for glycine. A point mutation happens. Now we have AGC, which codes for serine. Serine is chemically different from glycine, so that can affect how the protein folds, and the protein may not fold correctly and will not be functional. Or maybe this amino acid is in the active site of an enzyme, and this mutation now prevents the substrate from binding. If this is a critical protein in a critical metabolic pathway, that organism will die. But it's possible that that protein is even better with this mutation. and that gives that organism an advantage. So it's possible. It's not likely, but it's possible. Another point mutation involves changing the codon from one that codes for an amino acid into a stop codon. We call that a nonsense mutation. So a codon that normally codes for an amino acid becomes a stop codon. And so that will result in a short protein. It is very unlikely that short protein is going to be functional or be able to do anything. And again, if it's a critical metabolic pathway, that organism will die. A frameshift mutation is one where a nucleotide is added or deleted, and after the addition or deletion, all the codons after are completely different. If you add or delete a nucleotide, then it changes all the groups of 3 after that addition or deletion. So all the amino acids will be different. It's extremely unlikely that protein will serve any purpose because it's got lots of different amino acids in it now. So again, if it's a critical protein, that organism will die. But it's possible, very unlikely, but possible, a brand new protein gets created, which is so wonderful and helps that organism to survive that it's beneficial. So it's possible, just very unlikely. But those are the common mutations in bacteria. We don't really talk about inversions or translocations because that's not normally an issue with the circular chromosome that bacteria have. When it comes to ionizing radiation, a very important mutagen, because ionizing radiation, very high intensity radiation, very penetrating, it goes right through cells and does a lot of damage to DNA. It causes free radicals to form. And so too many X-rays or gamma rays are lethal, right? They'll kill. But we all live on a planet, floating through a universe which has X-rays and gamma rays. And of course, there are substances here that naturally emit X-rays and gamma rays. And so all the organisms on the Earth have evolved to deal with a little bit of exposure to ionizing radiation. So we do have DNA repair enzymes. But you can only repair a little bit of damage caused by this radiation. So a massive exposure or repeated exposure to things like X-rays, you don't want to get an X-ray every month for the rest of your life because you'll get cancer from that. But a small amount, we can repair the damage. But too much and we can't. Same thing with UV radiation. So UV is not as penetrating as X-rays or gamma rays. So UV is non-ionizing radiation, but it does cause a problem in DNA. And the problem is if you have 2 thymines side by side, UV light excites them and causes them to chemically bond to each other. And they form a thymine dimer. The problem is that DNA polymerase and RNA polymerase, when they encounter the thymine dimer, they do not add the correct complementary adenines. So a mutation occurs. Now, all the forms of life that have evolved exposed to sunlight have DNA repair enzymes, which can cut out the thymine dimines and replace them with 2 regular thymines. But again, it depends on the amount of damage and over how long a period of time. You can kill bacteria with ultraviolet light. And we'll talk about an experiment in the future where we do that, and you'll see how lethal UV light is. But many of us have already experienced this. If you've ever gotten a sunburn, especially if your skin blisters and peels, UV light has destroyed those epithelial cells. And unfortunately, we do accumulate a lot of UV damage during our lifetime. And we get a lot of it when we're young and stupid, right? Because we don't know any better. And then as we get older, we start wearing sunscreen and minimize exposure during the worst time of the day and things like that. But there is some benefit to UV exposure. And what's that benefit? What's the benefit of some UV exposure? Vitamin D, right. So UV does help to produce vitamin D in the skin. But if you're light-skinned, you only need about 10 or 15 minutes of sun exposure on your hands and face to make all the vitamin D you need that day. But the darker the skin, the more exposure you need to UV to make the same amount of vitamin D. But there is some benefit to UV. It's just that you don't need to spend a lot of time out in the sun. So is there such a thing as a healthy tan? Although tanning season is over for this year pretty much, is there any real benefit to getting a nice dark brown tan over the summer? No. But don't you look at some young woman who's nice and bronzed and think, wow, she looks so sexy. Of course, by the time she turns 50, she won't look sexy anymore. She'll look like a leather handbag, right? And we've all seen these people, right? Too much sun exposure, and their skin gets very leathery and they're very wrinkled, and that's caused by the UV damage. So if you're just a little bit tan by the end of the summer, yeah, that's fine. A little bit of UV exposure, most days, but you do not want to lie out in the sun and tan. And you absolutely don't want to pay someone to irradiate you in a tanning salon, right? Because too much UV is bad. It causes damage, and that damage is cumulative. And it increases your risk of skin cancer the longer you live. So a little bit of UV is good, and too much is bad, right? Now, we can also use radiation to increase the shelf life of food, because if you irradiate it, you kill the microorganisms which cause food to spoil. So now, the radiation does not make the food radioactive, so that's not the problem. But there is some controversy associated with irradiating food. And so the radiation, it can cause free radicals to form in the food, and then you eat those free radicals. It can also potentially destroy nutrients in the food. So there is some controversy associated with irradiating food, but it does not make the food radioactive, but it may reduce nutritional content and increase free radical formation in the food. But it does significantly increase shelf life, right, by destroying microorganisms that would otherwise spoil that food. So any questions or comments?
So, Professor, the second one, are you saying food should not be radiated?
I'm saying that it's controversial. So it depends on your point of view. If you live in a region of the world where it's very hard to preserve food, then it might be beneficial to irradiate it. That way it can last a lot longer through shipping and distribution. But it may have lower nutrient content. It may contain more free radicals. So it is controversial. Any other questions or comments?
Professor, please, your PowerPoint, where can it be found?
On Blackboard.
But is it the one on your website?
No, on Blackboard.
Can you please share your screen on how to get there?
Yeah, let's share the screen here. So on the Blackboard page, you'll see, let me switch this is what you see. All right, course content.
Mm-hmm.
And then like for week 5. Let's see. Oh yeah, also yeah. You guys just have lecture notes, right, for micro. So you'll have the lecture handouts. And then in the recordings are the PowerPoints. So when you go back to watch this recording, that's where you'll get the PowerPoints.
Okay, thank you.
All right?
Thank you.
Mm-hmm. Okay, so if there's no other questions, then we'll stop lecture and we'll start lab at about 10 after 1. Okay?
Okay, Professor.